Good afternoon, everyone, and welcome to the Vegetable Education Conference. I'm Hugh Moy, and I'm the A&R agent in Decatur County, and we have Macy Wheeler from the A&R agent in Decatur County, in Turner County, and we will be your moderators for this afternoon. And first, we'd like to thank WorkSource Georgia for sponsoring the 2025 Vegetable Educational Conference. And we have a big thank you to the Georgia Vegetable Commissioners. Most of the research presented has been funded in part by the Georgia Commodity Commission for Vegetables, and these are your dollars at work. I'd like to introduce our first speaker, Nick McGee. He's going to be presenting the overview of Georgia NRCS perspective on pollinator habitat and the role plant materials can play in improving habitat quality. He is from the Jimmy Carter Plant Materials, and he's the center manager associated with the USDA and NRCS. Nick?
Thank you, Hugh. Good afternoon, everyone. And so, like Hugh said, my name is Nick McGee. I'm with the NRCS Plant Materials Program and manager of the Jimmy Carter Plant Materials Center. I'm here today to talk with you about the perspective of our agency, NRCS, and pollinator habitat, and sort of what we think pollinators are, what we think the role they can play, and then a little bit there at the end about the role that my program, the Plant Materials Program through NRCS, maybe can play as we work through some of the challenges that pollinator habitat pose, especially in the southeast. So just a real quick overview of sort of what I'll talk about today is sort of a real quick introduction to what pollinators are and maybe the roles that they play for us in crop fields. We'll talk a little bit about what makes up good quality pollinator habitat, sort of the components of it. And is that better? And then we'll talk at the end about working with USDA and NRCS specifically and sort of the role that we can play at the Plant Materials Program in some of these issues with pollinator habitat. And so when we talk about pollinators and when the USDA talks about pollinators and what it is, you know, it can be any number of things. So, you know, there's bees, flies, wasps, butterflies, even up to, I saw recently that there's a fox that they've decided is a pollinator of certain plants now. But overwhelmingly, that functionality that's carried out for us is done by bees. And it's important to recognize that the pollination that we so rely on is actually a byproduct of them and their actual life cycle and what they're doing. They're feeding on the bloom and the nectar and getting nectar and pollen out of the bloom there, and the actual pollination that occurs in the plant is just sort of a byproduct of that. It's estimated that somewhere between about 75% and 90% of flowering plants on the globe require some sort of biotic pollination, and somewhere over 100 crops that we grow in the U.S. are relying on some sort of pollination in order to produce. It's hard to put a number on that, right? And so that the value that is assigned to it is somewhere in the tens of billions annually that we can assign to just in agricultural value. And when you look at the value to our environment, and you know, if you start drawing those lines where the bee pollinated the flower, which then the seed fell and fed the next line and so forth in the ecosystem, the value there is really incalculable. Some researchers a while back in 2015 looked at sort of the value of biotic pollination to Georgia crops and estimated it to be at somewhere around $367 million annually that's provided to us by pollination. And it really even occurs on some plants that you wouldn't think about and some crops that you wouldn't think about, including things like forage seed, vegetable seed, and some others, you know, fruits and nuts, and those where we see the bees or honeybees brought in is pretty obvious. But when you start thinking about seed crops and some of the value there, you can see where even something like the dairy industry may rely on pollination as well. And it's more than just the European honeybee. You know, it's real easy to think when we see these boxes, we think about on the side of the road, where the beekeeper has brought bees in. They really think that that's what we mean by pollinators, but it's really more than that, right? So there's roughly around 4,000 species of native bees in North America, and many of those do provide some level of pollination services to us. And the challenge is that in most cases it's difficult to differentiate between the two. So because the honeybee comes and goes in a flower, and then a native bee is right behind it or right before it, it's often difficult to determine which visit meant that the flower got pollinated. And then there are some situations where there are specialized species. In the case of the one there on the in the middle, where it's actually the southeastern blueberry bee, or in the bottom right there where you have the squash bee. Those are actually specialized species that are particularly good at pollinating certain plant species. And so when you start looking and comparing managed versus wild bees, there's no doubt that the bees that are in that box there that come home every night and are easy to pack up and move down the road certainly have their advantages, but the wild bees actually can provide some real some extra services to us that honeybees do not provide. And so researchers have looked at this and tried to put a value on just the pollination that's provided to us by wild bees. And they estimate that somewhere around in 2006, they estimated that somewhere around $3.1 billion was being provided to the agricultural industry just by wild bees alone, not including honeybees. And so, and also, if they looked at tomato as an example there, and in that case, there's a process called sonication, where there's a certain bee that goes in and can actually shake the tomato plant and pollinate it. And in that case, they were able to attribute a 45% increase in cross-pollination and a 200% increase in fruit weight just from that wild bee alone, not including the honeybee. And so then if you look at the number of bee visits, they actually will see that in most cases only around 25 to 30% of the bee visits to a flower are actually carried out by honeybees, and the other are carried out by wild bees in nature rather than the honeybees that are brought in. And so pollinators, as you might imagine, have become quite important to USDA, and so USDA has recognized how critically important they are to the agricultural industry, the environment, and to humans in general. In 2008, they actually went in and to the Farm Bill and prioritized pollinators in the Farm Bill through a number of different practices, including conservation practice standards through NRCS, and focusing additional money there, right? So in 2018, they expanded that look into pollinators in the 2018 Farm Bill, and started funding grant opportunities, encouraging collaboration between USDA agencies across the board, as well as promoting IPM practices and some other things to support pollinators, right? So then the question becomes, if we've decided that these wild bees in particular are valuable to us, how do we build habitat that is of good quality and makes good quality habitat for them? And so the Pollinator Partnership has identified that many of our pollinator populations are actually being threatened by habitat degradation and fragmentation, and they go on to list, pollution and pesticides and pathogens and a number of other things. But really, that first sentence drives it home, right? So it's this habitat degradation and fragmentation that's really driving our decline in native pollinators. And so the Xerces Society has gone a step further there and sort of identified these three basic needs that need to be met for these wild bee populations to thrive. And really we need to provide them with adequate food sources throughout the year. We need to provide them with safe nesting sites and access to clean water. And then we need to protect them from off-target pesticides. And so when we talk about, if we break that down and we start looking at habitat, right? So the first first thing there is we need to feed them, and we need to feed them throughout the year. And so the diagram that I have here is actually looking at apple and pear, but it's important, but the concept that is shown there by the diagram holds true no matter which crop you're looking at. But the point here is that the cash crop alone doesn't provide enough nectar and pollen to feed this native bee population throughout the year. And so you can see that if you look at those scales there, you can see that the bees are actually out and active for several months on end, and that the bloom period of the crop only is available there for a short period of time. So it stands to reason that we need to provide them with additional sources of food, pollen and nectar mainly, bloom periods, throughout the year. And really this need is almost year long. And this diagram was put together from bees in the Northeast where it's quite a bit colder. And you can see that even there they have bees active from March through November. I would say in the deep South it's probably extended even longer than that. It's also important to note here that when we talk about managed bees versus wild bees, managed bees have the advantage of being mobile. So when this bloom winds up here, in this case for the crop that's shown there in red, we can pick those bees up, move them somewhere else, and put them on a different blooming crop, allow them to continue to be fed. In the case of wild bees, where we're trying to grow habitat in place, we need to extend that bloom period out for longer. So what does that look like? So this is sort of the same idea, but now we're looking at the bloom time of all these different plants and how can we extend that time and keep these bees fed for a longer period of time. And so, excuse me, so really what we need is a diverse source of blooming plants throughout the year. And so you can see where the crop falls into that in this case apple bloom that it's showing, but we need some things that are blooming earlier than that to really promote the pollinator populations before we really are going to call on them and need them in our vegetable crop or fruit crop. And then we certainly need things after bloom to get these bees and these wild bees and other wild pollinators fed throughout the rest of the year so that they can overwinter and be of value to us the next year. And really when we look at these plants, USDA has taken the approach that really native plants tend to provide the best options there. Native plants typically tend to be well adapted to the region that you're growing them in. They typically are low maintenance, which is important when producers are interested in growing fruits and vegetables. They don't necessarily want to have to be putting a lot of maintenance into this habitat. And then they typically are perennial options. So once we get them planted and established, we don't necessarily have to rely on them to come back and need management again and again and again. There are some host-specific interactions with some of our native plants and certain pollinator species, so certainly that can be important as well when you look at natives, making sure that we have the right native plant there to host the right native bee. Non-invasive and non-noxious introduced plant species can be used in these habitats in some cases. It's really a case-by-case basis, but we tend to try to lean towards natives when possible just for the ease of use and low maintenance. So once we get them fed, then we have to really worry about their nesting habitat, right? And so when we talk about native bees in particular, with managed bees, typically their nest is in a box, and it's a box that's mobile and can be moved around. But with these native bees, that's not the case, and so we have to be able to provide them the home that they need. And so you have actually tunnel nesting bees in the case of the top left there. You have cavity nesting bees in the top center, and you have ground nesting bees in top right. And an interesting example of that, of a tunnel nesting bee, is this there on the bottom right, the mason bee, which actually goes in these tunnels and puts pollen and nectar, or excuse me, a mixture of pollen there, lays an egg on it, and then caps that cell off and moves further out. And that's how it rears its young. It's put enough food inside that cavity for that egg to hatch into larvae and pupate and become an adult before it emerges. And so once you get into this, you start thinking about habitat and habitat enhancement, you recognize that nesting sites and providing nesting sites for these native bees is actually just as important as providing food and blooms for them. And so So the way I look at that is it's just important to have the picture there on the top left as it is to have the one in the top center. And both of those are required for these species to really thrive and habitate in where we have asked them to be. So field borders and woodland edges and non-crop areas and all these things really are the type areas that we're looking for, the areas that we want to promote. And we really want those habitats to be in somewhat close proximity to the crop fields that you all are managing so that the bees can move back and forth between them, provide you the pollination service that you're after. Well-drained sandy soil tends to be really good for ground nesting bees, and so that really makes our southeastern fields with our small field shape and our landscape where we have small fields with a lot of woodland edges and non-crop areas scattered across the landscape really makes it as a good fit for nesting habitat for these native pollinators. And so it's important to leave that nesting habitat whenever possible. I mentioned that one of the things that has been attributed to the loss of pollinator habitat is fragmentation of the habitat itself. And so whenever we can leave this pollinator habitat in these non-crop areas undisturbed, that really helps in continuing the next population. And so it's best practice to only disturb about half of that habitat within a given year. It's best practice to leave woodlands intact nearby so that these tunnel and cavity nesting species have a place to go. And then it's also best practice to apply these management practices into the springtime after the bees have already emerged from the winter and started to be able to go out and forage and move around across the landscape. And lastly, I won't talk too much about it. Dr. Singleton may talk about it a little more in her talk, but about protecting these bees from pesticides. And so, it's just important that we give them a safe spot to go to. We keep use the least hazardous formulations as we can whenever possible. We follow the label, obviously. And really what we're after is what you see there in those pictures where we have fields that you all can manage however you need to, use the tools that you need, but then we have these sort of pollinator reserves there along the field edge that are protected from any of the applications that may have been made inside the field, the crop area. So if you go ahead and you talk with NRCS, how do you work with us on pollinator habitat? And so NRCS does provide both technical and financial assistance to work and establish pollinator habitat. We can actually identify and manage existing pollinator habitat on the farm, or we can create, enhance, and restore habitat that's not there yet. We do that through wildland or wildlife assessment guides. We come out and take a look and sort of evaluate what's there, what can be brought there, and it's important to know that it doesn't just have to be pollinator habitat. You can actually do any number of these practices, field strips, field borders, tree and shrub planting and sort of focus towards pollinators if you choose. For the sake of time, I'm going to skip on through this one pretty quick, but the idea here is that this is sort of that on-farm assessment. Xerxes, NRCS uses an in-house assessment, but Xerxes has one online that you can find there at the link. It's really just an idea of is this a suitable site for these pollinator habitat to be installed, and then can we meet those three basic needs that we talked about earlier in the talk. So the challenge here for the Southeast is that there's just really not a lot of information about the pollinator native species, the native species that are hosts, and the supply of, there's really a limited supply of southeastern ecotypes of plants. And so in often cases our producers are being forced to use non-local ecotypes when they go out and plant these natives, and they wind up with something that looks a lot like what's there on the right, which is a stand of crabgrass, basically, rather than what's there on the left. And so this is really where my program comes in, the plant materials program, is we're starting to focus headlong on some of these challenges and try to move that along. And so you know, I did this talk a little bit backwards, and so I'm introducing what my program is here at the end of the talk rather than at the beginning of the talk, but this is really where we fit in. And so the plant materials program is actually out there to assemble and test and release plant materials and plant technologies for conservation purposes. And over the years, the program that I work in has actually worked to release a number of conservation plants, some of them that you may be familiar with there, but a lot of native grasses. But that really is where I think this program can change and move towards supporting the pollinator habitat that you all are so interested in. So historically, we've worked on more of the management side of things, looking at management techniques on the plant stands and how does burning versus disking versus mowing fit and maintain these stands long term, or you know, how do we get these established? But really where I think we need to move to, and where my program is rapidly moving towards, is the release of new pollinator plants and making those available in the marketplace. The program has a history of doing that. In fact, you can see a number of these here that have been released from other centers that do support pollinator habitat. And so this is really where we're going to move to in the future, is selecting for plants that are southeastern ecotypes that foster and serve as hosts for the native bee species that we're interested in. So just to finish up here, there is a native seed network that has been stood up in the last couple of years that's going to foster this. And so this has really been a combination of a number of partners. You can see there on the left, it's led largely by the State Botanical Garden of Georgia at UGA. NRCS is certainly involved, but really it's a number of partners that are out collecting seed and finding southeastern ecotypes of these natives. Pollinator habitat is not the only focus, but it is one of the focuses, and it's really the vehicle that we're going to use to collect and maintain, or collect and release seed for this purpose. We go all the way. This project will go all the way from wild seed to commercial plant release. So we'll go from all the way from collecting seed in the nature through all of the increased activities and the selection processes, and then hand this seed over to a commercial seed grower at the end and ask them to provide it to you all. And so that's really what I've got time for today. I think I'm out of time, and so I'm going to stop right there. I don't know if I've even got time for a question. A couple questions, if there are any.
If there are any questions, I can bring the mic to you, or you can come to the middle.
No? Okay.
Thank you. All righty. Well, up next we have Dr. Taylor Randall Singleton from the University of Georgia. Thank you, Taylor.
Is the clicker working? Is that how we're doing it? Good afternoon. Thank you for that introduction. As she said, my name is Taylor Randall Singleton, and I am an extension specialist at the University of Georgia, where I focus on sustainability and agronomic and specialty crops, particularly surrounding pesticide stewardship. So I'm really excited to share with you today some of the research that we've been doing over the last couple years. And I'd like to recognize my co-authors and co-collaborators on this project as well, Dr. Stanley Culpepper, and then Nick McGee and Richard Barrett at the NRCS Plant Materials Center. And so it's a really exciting research project, a little bit different from the traditional row crop, traditional specialty crop, but something that's extremely important to our industry. Nick touched on it really well, so I'm not going to go too far into it, but I think we all recognize the importance of pollinators to our agriculture production. I saw some really interesting statistics when I started digging into this research a little bit. Over 30% of the world's food is pollinated, over 130 fruits and vegetables around the world, but really 1 in 3 bites of food that we eat, if we have a healthy diet, actually is pollinated by an insect. Why should we care from a production agriculture standpoint? Well, I think it's probably very familiar to you and your operations. But across the U.S., there's over $27 billion a year in annual income that comes from crop yields that are serviced from pollinators. In Georgia, that's over $600 million alone. And these services from pollinators are absolutely free to us. And so it's really, really important that we do what we can to support these insects moving forward into the future. But beyond pollination, beyond the service that the pollinator provides, so of course without pollination we have no seed, we have no fruit, we have no yield, but what else should we consider with our operations? Well, I think that there's really 3 different categories that pollinators bring to our row crop and specialty crop operations. That's environmental and ecological stewardship. I think there's an opportunity for positive public engagement. And then, of course, the issue that really has drawn my attention to pollinator habitat is the implications that these habitats have on pesticide stewardship moving into the future. And so first, looking at the environmental and ecological stewardship portion of a pollinator habitat, of course, pollinator habitats provide biodiversity to the landscape. Insects like a diverse diet, just like we do. But there's also a really unique opportunity to support wildlife habitat on our farms. This is actually from some of my research plots this year. The first time in my life I've ever seen a quail nest. So really unique opportunity to bring other wildlife onto our operations and support these populations as well. Nick talked on this a little bit, so I won't get into it too much, but there's an opportunity with pollinator habitats to support these insects year-round by nesting habitats and just providing them a safe haven to continue their life cycles. There's also a really unique opportunity, I think, to engage the public in a positive manner. When we're talking about production agriculture, specialty crop production, any opportunity that we have to bring the public in and share what we're doing on the farm is an opportunity that I think we should take. Pollinator habitats are one way I think we can just bridge that gap. They're very eye-catching, especially when they're in full bloom, and really just provide a unique conversation opening to what's going on the farm, what are you doing, and just a way to engage those that aren't actively involved in agriculture. And I think pollinator habitats are a really neat way to do that as well. Well, what I'm most interested in is the use of pollinator habitats when we start talking about pesticide stewardship. And I know that's 2 things that usually aren't in the same conversation, but it's really an issue that is plaguing our industry moving forward and we have to have on our radar. Of course, pollinator habitats is really, how do we measure that return on investment? Well, it's really hard to quantify, of course. We don't directly increase yield that we can measure very easily. They're an extra expense to install and maintain on our operations. But one thing that pollinator habitats have direct implications on moving into the future is the ability to support endangered, listed, and sensitive species as we talk about some of the new pesticide regulations that are being placed upon our industry. If you weren't in the ESA session this morning and aren't familiar with what's going on regarding pesticide regulations and the Endangered Species Act, get with me afterwards and I'll catch you up. But pollinator habitats have the ability to support the continued use of pesticides moving into the future, as crazy as that sounds. And I think that there's a real opportunity that we can work together on this. And of course, there is an opportunity to use these sites as a way to mitigate runoff and erosion of a pesticide product from a treated field into one of these sensitive habitats. And we'll talk about that a little bit more. There's a lot of support right now at the state and national level regarding cost share and programs to install these habitats on our land. NRCS is doing a lot of funding right now to install these habitats and that would benefit pollinators on working farms. There's actually over 30 different programs right now where growers can receive assistance, cost share to implement these sites on their farms. A couple of these options are the habitat installation programs themselves, working with field borders, riparian buffers, filter strips, native grasslands. All of these options are available through NRCS and there's funding at the state and national level. But what is really interesting to me and what really ties back into that pesticide stewardship component is that these practices that are funded through NRCS to supplement pollinator habitat also directly tie into the new herbicide strategy, which is the EPA's framework for evaluating pesticides to ensure they are in compliance with the Endangered Species Act moving forward. Again, if you want to catch up on that a little bit, I'll be glad to talk to you afterwards. But these programs can really help farmers ensure that they are in compliance with this new strategy moving forward. So looking in at the NRCS programs at the state level, so this is specifically for Georgia. Every state may be a little bit different, but for Georgia specifically, if you want to enter one of these NRCS programs, there's standards that have to be met when you're implementing your pollinator habitat, and that includes what diversity of species need to be present in these sites. So in Georgia, we need 9 broadleaves, we need 1 native grass, we need a full season bloom period, and we need that site to persist over years. We have heard from growers through extension that this is extremely hard to achieve on their land. These plants, many of the native species have a low emergence, they are not competitive, and when we start talking about putting these sites on active and productive agriculture land, you have to consider the weed pressure that we have. Growers have told us that based on the extensive weed pressure that they have on their lands, having these non-competitive plants on there, it really just doesn't ever establish. And so that really caught our attention. And so let's say you want to plant a habitat on the farm. You have an unproductive, you have a bare area of your farm. Can you just throw out some seeds, some pollinator mix that you buy at the store, and will it work? It's not recommended, and it is going to cost a lot of money. From what we've learned, there are many challenges and many questions that have to be answered before you actually go out and plant that pollinator habitat. You need to have a good plan in place. You need to understand what species are going to be compatible with your land. How do you plant those species? How do you maintain those species? But most importantly, if you are going on to a piece of agriculture land where you have extensive weed pressure, how do you control weeds in that site so those plants actually have a chance of coming up, emerging, and getting to the point where they are growing, they're competitive, and they can maintain over the years? And so at University of Georgia, we are really interested in how can we help bridge this gap. Pesticide stewardship is extremely important to us, but we also want to support these pollinators, these sensitive species. So how can we do this in a way that is going to be very efficient on the farm? It's also going to be successful. And so our goal is to help growers implement pollinator habitat on their farm. We wanted to investigate a couple different questions when we started into this project. I think the first year was in 2022. We wanted to look at a mixture of different native species that were suitable for NRCS programs in Georgia. We wanted to see how do we control weeds during this establishment period. Those plants have to have a chance to get up and growing. We have to be able to control weeds so that they have a chance to actually come up and do their thing. And so we wanted to look at different pre-emergent residual herbicides to see if we could keep the weeds at bay so those plants, those native species could come up and grow. And then we wanted to screen several different post-emergence herbicides so that we could actually go in and clean up any weed issues once those plants were growing if we needed to. And then we also wanted to evaluate persistence over time. And so in the first year in 2022, we started work at the University of Georgia Ponder Research Farm in Ty, Georgia. If you're not familiar, this research farm has to have the highest weed population in the state because this is the Weed Science Research Farm. And so we knew that we were really going to put these herbicides to the test and see what was going to happen with our native species. And so we were overly ambitious our first year and decided to screen 23 different native species for Georgia. We had overhead irrigation available to us. We hand weeded all of these plots, so we had no weed competition. That allowed us to really see what the herbicide itself was going to do to that native species. Did we have true tolerance? Because there was no competition to that plant. We looked at a variety of different pre-emergence and post-emergence herbicides, and we had a non-treated control available for every species. And so ideally, we had the perfect planting and growing conditions available. We use this piece of equipment here on the left side of the screen. This is a mini vegetable seeder. So we put that seed exactly where it needed to go, anywhere from 1/8 of an inch to 1/4 of an inch in the ground. Perfect growing conditions. It was an absolute disaster. Our first year, we had very, very little emergence. We learned a lot that first year. You can see on the left, we have our seeding rates here, but our emergence rate in most cases was less than 1% for each of these species. And this is at multiple different sites. And so what exactly had gone on? And so we started digging in a little bit more to really understand these species better. We revisited our initial objectives and we scratched the whole thing and decided to really switch our focus going into the next year. If these weeds are so competitive and these native species themselves are so sensitive, they're non-competitive, they really just need to have a chance to get up and get going before they can become serviceable to the insects. We needed to give them that opportunity to do that. It's really hard hard to do on a production agriculture farm where weed pressure is extremely intense. And so how do we keep that ground covered so that those plants can have a chance to come up and get blooming? And so we switched our focus that second year, going into more of a, what if we started with more commercialized annual species, something that we know is fast growing, it's hardy, and there may be some herbicide tolerance in there. Can we get those species up? Can we get them going, have them cover the ground, which would allow us to have an opportunity to add these native species in the fall? And so we started with 4 different annual species that are commercialized, zinnia, Cosmo, marigold, and Mexican sunflower. We looked at a variety of different pre-emergence herbicides to really just broad spectrum screen many different products to see if we had tolerance in those 4 species so that we could plant those species, have a pre-emergence herbicide, keep the weeds down, and allow those species to get up and bloom. And so in 2023 and 2024, we screened a lot of different pre-emergence herbicides, really focusing on what products are available to help us control a broad spectrum of weeds that may be present on a production agriculture scenario. And so here's the list of products that we screened over the last couple years. Again, we were looking at tolerance within zinnia, Cosmo, marigold, and Mexican sunflower. We planted each of these species with the same vegetable seeder. We had a very similar scenario, perfect growing conditions, hand weed to keep the competition down, and really saw some interesting results. And so from our screening, we found a lot of options that were absolutely not going to work. And so in the red box there, you can see these herbicide options were not compatible with these flower species. They really were detrimental to the species themselves. Severe stunting, most of them killed those species. We had a couple options that were maybes. Maybe these herbicides could work as a pre-emergence residual product if we play around with the rate a little bit. And then we actually had several products that looked really good as far as species tolerance to those particular herbicide products. We looked at, does this product cause injury to the flower species itself? What does it do to the height of the species? Does it influence the stand of the species? Really important when we start talking about ground coverage. How does it influence the ability of that plant to go to bloom? It's really important to understand that we don't want to delay bloom of that flower plant if a full season bloom period is critical. And then we also looked at bloom counts. So really interesting, just look at broad spectrum. Are there herbicide options that are available that we can use to control weeds during the establishment of these particular flower species? And so being trained by a weed scientist, this got me thinking, we have to remember the principles of sound weed management. They still apply even though we're talking about a flower species. We're not talking about a vegetable here, but we're talking about a flower species. These principles of weed management still apply. If you've been coming to this conference for years, you understand Dr. Culpeper has been teaching us for years. We have to take a systems approach when talking about weed management of a crop. We want to be diversified in our weed management approach. We want to be timely, and we want to be very smart. And so thinking through what is it going to take to keep weeds at bay so we can get flower species up. We need to be as clean at planting as possible, no weeds present when we actually put the seed in the ground. We want to have pre-emergence residual herbicides that will allow the actual flower species to come up, keep the weeds down. We want to be able to use post-emergence products if necessary in order to keep that site clean, and we need to manage the grasses in these sites if possible. And so moving into year 3 of our research, we decided to take a different approach. Using the information that we learned from our herbicide screening studies, we wanted to look at a systems approach of weed management. So how do we actually use tank mixtures of different residual herbicide products to control a broad spectrum of weeds that may be present in our site? But then also, how do we actually plant these species to see if we can get optimum ground coverage in order to use that to our advantage to suppress some weeds that may come through? So I know this is a lot of data here and the text is really small, but I just wanted to use this to illustrate a point real quick. It's really an interesting project, an interesting approach when you start looking at a systems approach to weed management. We want to use multiple herbicide products to control a broad spectrum of weeds that may be present in our site. And so that's exactly what we did. We pulled the herbicides that look good from our pre-emergent screening study, and we just put together different combinations of different products to see if we could find something that when we put that tank mixture onto the soil, when we put those 2 products together, we don't have excessive injury to that crop, to that flower itself. And so having 30% injury as our threshold, beyond 30% injury, we saw issues with stand, we saw issues with plant height and bloom period. So setting that injury threshold at 30%, we actually had a couple options that looked pretty good as far as keeping weed emergence down with a tank mixture of products that could provide a broad spectrum of weed control throughout the year. And so this leads us into 2025, where we're going to actually expand on these tank mixtures, look at the different products that look pretty good in the first year, look at different application rates, and see if we can really dial in on how to control these weeds using these tank mixture products. But as far as managing weeds in the site, of course, it's beyond just applying the herbicides. We have to consider, can we keep weed pressure down by influencing or impacting the actual planting of the flower itself? How can we use that to our advantage? And so we actually created a mixture of these 4 species and spread them across the site using modification to our grain drill. We actually just opened up the tubes there and let the seed fall to the ground. We wanted to look at how can we influence ground coverage using planting method. And so we then did a light cultivation to incorporate the seed and compared broadcast seeding versus spreading the seed with the grain drill. It was really interesting. So when you look at broadcast versus drilled, the drill, using the grain drill to plant the seed actually resulted in 25% faster ground coverage compared to broadcasting the seed. So really providing an opportunity to use planting method to influence how fast ground coverage occurs, which influences our ability to suppress weeds longer throughout that establishment period. We also found when we looked at different combinations of the species themselves, so different ratios of that seed within a seeding mix, we found that using faster, more aggressive growing species at a higher ratio compared to the other species allowed us to have a quicker ground coverage as well. So different options available that allow us to really influence the ability to get that ground covered quicker and suppress weeds. We also found that looking at planting method itself, so again, broadcast versus drilled, influences how quickly we see blooms in that first growing season. With the drilled, using the grain drill to plant our seed, we found that we had 25% faster blooms, which means more time for pollinators to be out at that site and then servicing the cash crop itself. So also in year 3, we moved into a collaboration with NRCS, so with Nick that just spoke. We started working with him and his team at the Jimmy Carter Plant Materials Center, comparing what we're calling our UGA mix, the commercialized annual mix, the zinnias, cosmos, marigolds, and Mexican sunflowers, compared to an NRCS mix, a traditional mix of native species, and we had several different native species there on the right side of the screen. And so what we did was we just planted these species side by side. We wanted to compare how fast do we see ground coverage of the species themselves, how fast do we see these species blooming, and do they persist over time. And so here's an example. I've just got a couple pictures on the next couple slides. This is our NRCS mixture from our first site. We included a couple different herbicides just to see if we could help keep these weeds down while the natives and the annuals were emerging. On the left side of the screen, we have the no herbicide. In the middle, we have a pre-emergence application of Treflan. And then on the right side of the screen, we have a post-emergence application of Cadre. You can see here we have a very healthy stand of wild radish, but unfortunately none of our native species were coming through at this time. Just being non-competitive and very slow-growing species, they didn't stand a chance against the very aggressive wild radish that was growing at the same time. But when we compare it against our UGA annual mix, this is early in the season at the same time, we have really good ground coverage of the annuals themselves, very little weed pressure at this time, and starting to see a few blooms. This is 51 days after planting. At our second site, same story. We had intense weed pressure. The weeds were outcompeting any of the natives that were coming up. Remember, most natives come up very slowly. They're spending that first year developing their root system to come through on the second year. And so just not competitive against, in this site, we had an intense population of palmer amaranth, wild radish, nutsedge. They just can't compete with these aggressive weeds at the time. However, with our annual mixture, we have many different blooms coming through at this time, really good ground coverage of the species, and very low weed pressure due to that suppression from these species themselves. And so few weeds, great ground coverage, but can we cover the ground to suppress those weeds for the natives to establish maybe in the next season? And so that's where we're going with this work. Can we use these annual species to get the ground covered in the first year? We have the blooms there for the insects to utilize. Can we use that to really provide an opportunity to get those natives out there? Maybe we go out and plant them in the fall, give them a chance to get going without that competition from the weeds, to move into more of a native seed mixture moving into the following years. And so that's the direction that we're going with our next location. Also really excited to work with NRCS on an on-farm project starting this year and moving into the next couple years. Working again with Nick and his team, we hope to evaluate pollinator and switchgrass habitat on-farm at various locations throughout the state of Georgia. This year we'll have 4 locations, mainly located in South Georgia. We hope to expand next year. But we really want to focus here on two objectives. How do we work together to establish pollinator habitat on farms? But how do we, going back to Nick's previous point, protect those pollinator habitats from any impacts from pesticides? And so while using these sites as a way to mitigate pesticide runoff and mitigate particle drift, we'd like to incorporate pollinator and switchgrass habitat really to work together to see if we can accomplish these goals. We want to observe these pollinator habitats over time, see how they do on the farm, and do they persist, and do they accomplish what we set out to do. And so the best way to do this is just a visual example. On the far left side of the screen, we have our crop field. We're going to go in next to our crop field and actually plant a couple rows of switchgrass, which grows extremely tall and extremely wide. We're going to use that to protect our pollinator habitat from any potential particle drift that may leave that field following a pesticide application. We're going to collect runoff as well from these sites and see, is this an opportunity to use pollinator habitat and switchgrass plantings as a way to mitigate particle drift and mitigate runoff and erosion from the treated field. So really excited to work with NRCS on this project over the next couple years and see how this ties into pesticide stewardship moving into the future. And so we all understand the importance of pollinator habitats to our farm. Protecting pollinators is critical to our farm sustainability, not only from an agronomic but from an economic perspective, but also environmental and a regulatory perspective when we start talking about pesticide stewardship and the challenges that pesticides face moving into the coming years. Excited to continue research collaborations with partners at USDA, NRCS, and UGA to come up with solutions that are practical for the farmers to implement on their farm. So at this time, I think that I'm close on time. So thank you for your attention today. I'd like to thank Tim Richards, Kaylee Roberts, and Sydney Grantham for their helps in implementing this project. And this research is being funded by the Weed Science Society of America, so we're really excited to work with them on that as well. Thank you for your time. I'll be happy to answer any questions if you have any.
Well, thanks.
You.
Here, this is for you.
Alrighty, we're going to go ahead and get started with the next session. We have Dr. Ruinito Ferrarazzi from the University of Georgia. And before we get started, just make sure you are signing in at the kiosk to get your credits and fill out the green sheets for evaluations, please, and just leave them. We'll pick them up afterwards. Oh, thank you.
Thank you. Good afternoon, everybody. If you don't mind, I will get the mic and I will move around. So the teacher in me asks me to move around a little bit. So we are going to talk about vegetable production under controlled environments. And I have a very great mission this afternoon. It's kind of mission impossible. I have 700 slides to cover in 20 minutes. Let's go with it. So I just want to bring this over to everybody. So my lab does a lot of activities because I work together with people and growers. So please use this opportunity to connect with me and to expand this even further if you see an interest in controlled environment. So I wanted to start by just bringing some of these growers to your attention. So I work closer with Pure Flavor, Local Bounty, and other growers. As you can see over here, a lot of companies have been working with me in the last 3 years. So I want to thank them ahead of time. I'm not seeing any of the people that I listed over here, so I will skip the names today. Even though I just saw Daniel Jackson in the back, who is a close partner from the AESL lab. Thank you for being here, Daniel. So, oops. My program is, oops. I'm going backwards on the number of slides. So my program had been fortunate to receive funding from all of these agencies, and through them, so the research that I'm going to show it to you is possible. I just have a few slides highlighting what I do on a 30,000 feet overview. 30% of my time is spent in teaching, so I'm helping prepare the next generation of growers, right, or crop advisors. So I do teach hydroponics and controlled environment agriculture, and these are some of the pictures of the activities we do in the greenhouses and the students we have in Athens. One former student here in the audience, nice to see you, Katie. And I also teach one course that is based in ornamental production, that is greenhouse management. So as you can see, beautiful plants, we tour a lot of different growers, and we also organize a lot of mixer activities with the industry. So the core of what I'm here to talk about today is my research program. So my lab is the CEA Crop Physiology and Production Lab, and I do quite a lot. So some of you will recognize a little bit of my past work. What I'm going to show it to you today is my current work. And my lab is quite vibrant, so I invite all of you to go and visit us when you have the chance when you are in Athens. All right, so before we proceed, we are in the Controlled Environment Agriculture section. Who here doesn't know what CEA is? Do you need any help trying to understand what Controlled Environment Agriculture is? No? That's great. So you made my life easier. I was just going to show it to you that what we consider CEA is actually everything that is produced under some sort of protective structure that allows us to control the environment. And since everybody here is highly knowledgeable on these particular structures, I just wanted to point out that I don't cover high tunnels in my research program. There are other colleagues at UGA that do that. So we focus more on mid and high-tech greenhouses and on the vertical farms, all right? And you can see that arrow. So as we go up on these particular names of structures, so what usually happens is we increase yield, we increase risk, we increase cost, okay? So the part that I like the most to share about is what I do, okay? So you are going to see here a list of projects that I have focusing on irrigation, focusing on plant nutrition, and also focusing on cropping systems. So these are the projects that I will talk about today. What I have listed in blue are the other activities we are currently doing at my program, and due to the sake of time, I will skip them, but I'll be more than glad to share those with you if you have the interest, okay? So that being said, I would like to start by pointing out that the breadth of my research program really gravitates around finding production tools to assist growers on their endeavors, all right? So let me start with one of our simplest projects that was basically a hydroponics system test coupled with a water management strategy that was done by a former graduate student that is now a Peace Corps in Mexico, Mr. George Hutchinson. What I'm doing for these projects that I'm showing to you, so I'm skipping the intro, the objectives, and I'm going to the nitty-gritty details of that particular project. So let's see if that works for you to understand a little bit of what we do. So as you can see here, we have a hydroponic system comparison under LED lights. And under this particular experiment, I tested 4 hydroponic systems. Everything that I'm showing is replicated, everything, is done for scientific publications. So what I'm doing is summarizing it for you. So we tested deep water culture system, a variant of NFT system, and we split that into a horizontal and a vertical. And over here, you can see a picture of each of the systems that we tested, a horizontal, a vertical, and an aeroponic system. All right, so everybody familiar with those systems? Wonderful. Okay, so 2 sets of graphs only. So in the top, we have yield. Over here, we have the 2 crops we tested, arugula on the left and lettuce on the right. And then here in the bottom, we have the 4 systems that we tested, okay? So what we can clearly see, big differences between crops concerning the results. We also see that over the treatments we tested, plants performed differently. When we expand these and look over the results concerning the efficiency of that system, so here in the top we have water use efficiency, in the middle energy use efficiency, and in the bottom area use efficiency, or yield per area. That is a square meter. What we can see, again, a clear, distinct difference between crops. There is also a difference between systems. So the bottom line message that I want to bring over here is that there is no one recipe fits all. So for each crop that you are growing, so you have to select the proper hydroponic system. So we have the capability to test it, and if you want further information about this particular result, so we can share with you later on. Because we have a similar system test with the strawberries, a hot commodity in the CEA world, particularly after the announcement of very large enterprises in Virginia. So what we see here is the same type of testing with the substitution of the deep water culture for a substrate system. So a picture here highlighting the plants. Sorry, I realize now that it's very hard to see the details, but as you can see, we have a dense canopy as we go down into these systems. So we tested the same ones as we tested before. And the results were similar to an extent, but I highlighted here something very unique, is the fact that the total fresh fruit yield was much higher in the substrate system as opposed to the other systems. And here I have 2 cultivars of strawberries. They're newly released Florida cultivars. And really bring over the fact that strawberries do not like water culture systems. They prefer substrate-based systems. The same type of graphs showed previously, so you can really see a spike in the substrate over here for water, energy, and yield per area. But we also have another winner into the system, which are the vertical towers, highlighting why the companies are investing in this particular system, because the yield per area is much higher than the yield per plant as compared to other systems. Okay? So one area that is very important for us is water management. So we tested the common systems that we have. Leaching fraction, that is the amount of water that's usually run off in excess. we tested soil moisture sensors, and we compared with timers. It's no surprise that the plants managed through soil moisture sensors or leaching fraction grew much better than the plants that were grown under a timer-based operation. So as you can see, it follows the same order. We have the leachate, we have 4 treatments with soil moisture sensors, and we had a timer-based treatment, which is the last one over here. And it poorly performed, even with the use of 2 different cultivars, highlighting that we need to be strategic on how we manage water in strawberries as well. Okay? So, sorry, I'm looking more into this screen, but the same is being projected on the other side, right? So I don't want to keep you dizzy from one side to another. Moving on, I did have another experiment looking not only into water, not only looking into systems, but looking into nutrient management. And this was done through a master's student that also just graduated, and we were looking into identifying optimal strategies for managing recirculating systems. We decided to go a little wild here, and we compared production systems under 2 different conditions. Vertical farms, as you can see over here, and our vertical farm had 3 increasing light levels, 250 micromoles, so standard, 500, which is high, and even a much higher level, 700, which had been used by some companies. We did the same testing in the greenhouse, used the same amount of light, and just a different environment. And what we also did was to increase, which was the critical point of this particular study, to look into nutrient management strategies. So how to manage the fertilizer solution replenishment. And we had 6 elegant strategies. Okay, I call that because we used the, yep, like magic, huh? Drain and refill, top off with full strength solution, with half strength solution. We also had another one where we added water and then we brought the EC back to a regular level. We also had a water use efficiency where we used the amount of biomass that grew, the amount of water used in the target that we had for the fertilizer solution we wanted to apply, and a very complicated system that we collected samples, sent to a lab, and replaced the nutrients on demand depending on which nutrient was needed in the solution. All right? So we went from non-targeted, simple, to targeted, complicated management. And the results were quite nice. So in the top, we have the 2 systems we compared, greenhouses and vertical farms. For lettuce, there was a significant difference between the vertical farm and the greenhouse. So vertical farms produced more than greenhouses. The graph here in the middle shows that as we increase the light levels, lettuce produced more biomass over time. So no novelty there. But when we look at the bottom graph, so basically it indicates that there were no differences between the different nutrient solution replenishment strategies, highlighting that growers can use more sophisticated systems without any compromise on yield, but saving nutrients and not releasing them to the environment. When we look at spinach, the results were similar, even though we did not see any difference between the greenhouses and the vertical farms. We did see the same difference in terms of yield as we increased light levels, but we reached a plateau at 500, indicating that there is no need to go beyond that very high light level. And the same applied to the different nutrient management strategies. All right, so really highlighting that one can be more efficient in managing the nutrients. Nutrient is not the only factor, so in this study we also provided a lot of different information to you if you wanted to move along with a more precise nutrient management, taking the pH drift, labor, cost, and risk on these two particular leafy greens. So I'm happy to share that with you if you are interested. Okay, moving forward. So 2 more to go. Studies, 650 slides still. Huh? We have a signature project that is to identify a methodology of sap analysis for CA crops. So this study is really looking into a comprehensive solution for growers interested in sap analysis, and it had been developed in partnership with the AESL lab and had been sponsored by the Georgia Department of Ag. And this student here, whose name Rauf, is the one working on his PhD for this project. Who here is familiar with sap analysis? You're not? So sap analysis, Tom, is like the blood test, of a plant. So we basically take the sap, send it to a lab, receive, a report with the nutrients that are present in that particular sap, allowing us to see what is taken up by the plant at that given time, and allowing us to really quickly, fertilize that plant, see that value going up, or cutting off fertilization, watering to see that value going down. So nice that you asked, because this is not a very common technique for CA crops. So I did publish a study about the process involved on it. We usually collect, extract, and analyze the sap, but there is no standard. Each lab have their own techniques, and they do not disclose what they use, for that particular extraction method. So we try to address that. And we conducted this study, excuse me for a sec, to basically test the most adequate extraction method, to identify the best sampling procedure, and establish sufficiency ranges to allow one to basically look into a table and figure out how much fertilizer to apply. All right, so this is a comprehensive study. Okay, so we tested 3 different crops and 3 different cultivars within each crop. As you can see here, so those are all modern cultivars. And they were chosen because we partnered with Local Bounty, Pure Flavor, and Calera, using their production facilities to sample these particular materials. And through strong support from AESL, we were able to get a lot of information concerning the methodologies that are available out there. So those are the ones in light brown. And we compared those methodologies with the standard leaf nutrient analysis method that is well calibrated. And our goal was then to identify which of these methods could be used by the AESL in future services for you growers. So we were very cautious on the stages. We did pick different leaf developmental stages over the course of the study, and analyzed leaf and petiole. And just a quick summary, the student was very diligent in picking a specific leaf, getting that leaf, chopping the leaf off in multiple different parts, subjecting them to random selection for analysis. So this is for lettuce. We did the same for tomato, and we went to analyze the leaf and also the petiole of tomato, and the same for cucumber. As you can imagine, so massive work. So this is a picture of the student working. He analyzed about 3,000 samples. So the idea is to create a very powerful tool that you can use it right away when you are ready for that, and when we release the results, of course. Everything was analyzed, and this was important for us to continue with the second phase of our study, which was to look into the number of leaves, when to collect those samples, early in the morning, late in the morning, when plants are not that turgid, the age of the leaves, the part of the tissue, and most importantly, the frequency of the sampling. All right, so as you can see, so if I was going to explain all the results to you, so this would be another talk. So I just brought this over to you because the idea was to highlight the opportunities that we have to use sap analysis for production. And we found that one extraction method does not feed all nutrients. So in fact, they were very different from one another, creating a massive issue for the lab that will be conducting this analysis. We identified that new leaves are preferred and that no differences in sampling frequency and time of collection. This is a picture of the third objective, the sufficiency ranges. All right, that is currently going on in a greenhouse in Athens. Okay, so I will save you from the details. So again, please come to me if you need any additional information concerning SAP analysis, because I'm ready to jump into the last one. Oops. 20 minutes already? Okay, all right. So I will have to skip the other 600 slides, folks. So I just wanted to indicate to you that we did a very interesting study with oxygen by adding dissolved oxygen into solution. And the results are outstanding, highlighting that we can really maximize crop growth depending on the crop we select based on the rate we choose to add into the water. But again, this is cultivar specific. So we had 2 arugulas and 2 kales. I will skip it. And I just wanted to bring over the same message to you. So be careful with one system that can be used for all crops that you have. That is not the case for the majority of the hydroponics production, okay? So with that, I would like to thank you all. And I'm here for questions if you want to talk to me afterwards.
Thank you. All right. Next up, we have Dr. Andrew Ogden. He's going to be talking about breeding vegetables for controlled environment agriculture production. He's the assistant professor and controlled environmental vegetable breeder from the University of Georgia.
Great. Thank you for that introduction. It's a pleasure to be here with you all today. So the title of my talk is Breeding Vegetable Crops for Controlled Environment Agriculture, which I do consider to be a new horizon, as it's a pretty new endeavor, as CEA is still quite new. I wanted to start with talking a little bit about how I ended up here today and a little bit about my background with CEA. I actually started quite a long time ago in CEA, working with, as what Juanito mentioned, as the simplest form of CEA, which are high tunnels. And we had a project where we grew blueberries inside of high tunnels to try to achieve early yields. Later, I moved on to the University of New Hampshire, where I had a teaching role. And while I was teaching at UNH, I also was able to conduct my doctorate. I had the rather unfortunate both of my mentors, my master's and my PhD mentor, have both passed away. But during my PhD, I really got exposed to plant breeding and worked extensively with cucurbit crops in New Hampshire. And then August of 23, I started at the Griffin campus. So my position is assistant professor. I have a split between research and extension. And my job is to try to breed some new cultivars of vegetable crops for CEA systems. And as you'll notice, you're going to hear from several of us that are all from the University of Georgia related to CEA. And that's because we do have a brand new team of faculty who are all dedicated to CEA, and we span several departments. And this was really in response to the rapid growth of CEA here in the state of Georgia. And I have some of the major firms highlighted here. But in addition to all these large operations, we have lots of small farms as well that tend to be more on the lower tech side, of the low tech greenhouses and high tunnels. But we are seeing a big expansion of CEA here in Georgia. So you might be wondering, why do we need breeding? Why does CEA require breeding? And the reason for that is currently most growers are just simply trying to adapt field-bred cultivars into CEA systems. And when we breed for field conditions, we have a lot of things that we have to think about that maybe are absent in CEA. Soil-borne pathogens we can largely eliminate in CEA. Drought stress we can largely eliminate because we're irrigating. And we can mostly mitigate temperature stress. So those are things that field breeders have to think about, but we get a bit of a break from by growing in these really optimized environments. So that enables us to start to breed for some attributes, crop attributes that are really unique and needed in CEA. So, although we can get rid of most pests through exclusion, we're not going to be able to eliminate them entirely. And small insect pests such as whiteflies, thrips, mites, these can all still pose problems. So we do still have to think about these pests. In addition, those insect pests not only cause direct damage, but also transmit plant viruses. And we still can run into problems with pathogens. So things like powdery mildew, different types of bacterial spots and wilts, and viruses can still be issues in CEA systems. So we don't get away from pests and diseases entirely. And so we need to make sure that we have crops with genetic resistance, as that's always our best tool for preventing pest and disease problems. And in addition, we need a greater diversity of crops. So far, it's only a handful of crops that have really proven themselves to be profitable in CEA, and we'd like to see a greater number of crops being produced. And as I've traveled to different conferences, CEA meetings, this need for breeding you hear repeated quite frequently. This was an article in Inside Grower magazine talking about the need for breeding CEA-specific crops. So one of the first things I did in my lab was to collaborate with several other breeders, and we have a new paper that's currently in press and should be out in about a month, and it is an open access paper, and that's called Improvement of Crop Production in Controlled Environment Agriculture Through Breeding. So a team of us worked together to publish this paper, and it runs through crop-specific needs for CEA. And to show a couple of figures from that paper that highlight some of these needs that are fairly unique to CEA. We had a couple of figures. Actually, Dr. Amol Nankar, who's here, contributed these figures to our paper. And these give you some idea about the types of traits that might be beneficial. You see a big fancy word there, phenotypic plasticity. And that's really the idea that, hey, could we get different performance out of plants based off of the environment that we put them in? But other things, amenable pollination and fruit set, using resources efficiently, again, that biotic stress tolerance. So these are some of the things that we want to breed for CEA. Now, fortunately, we have currently a wide range of tools at our disposal. So this is not as if we were trying to do this 100 years ago where we would be entirely dependent on just traditional methods. We have all kinds of new technologies and techniques that can be deployed to help us speed this process along to meet this need. So lots of different fields in here that can all be used in this quest to breed these new cultivars. So Overall, what you see there, we need to be able to, for CEA, we want to maximize our yields, we want to maximize crop quality, nutritional content, and flavor profiles in our CEA crops because we need to get a higher price for them to justify those additional costs. While at the same time, we need to find ways to reduce energy costs and other costs associated with CEA. And we're seeing, we have seen some large CEA firms fail in recent years. And in my opinion, that's due to inadequate research on the crops they're producing. We have to put a lot of thought into our crop selection, and any new cultivars need to be profitable in order to justify their production in these sophisticated systems. So some of the things that I've started working on, I have a project working on breeding some striped summer squash. We've seen cucumber take off in CEA, but not really squash yet. And so I've been breeding these to have a nice vertical growing habit, so not that ultra compact and bushy growth that you typically see on summer squash and zucchini, but rather more of a semi-bush or vining habit. And working on these different colors, so I have yellow, I have actually some green summer squash. Those are not zucchini, they're in the yellow summer squash group, but they're recessive for the genes that typically turn summer squash yellow. Striping genes in them just to help them really stand out in the marketplace as being very attractive fruits. And I'm incorporating powdery mildew tolerance or resistance into these whenever possible, because as soon as we put these crops into CEA, into greenhouses, powdery mildew inevitably rears its ugly head, especially in the fall and winter months. One of the great accomplishments of my mentor at UNH was identified a glabrous gene, which actually keeps these summer squash plants free of the trichomes that normally sting your skin when you go to harvest them. If you've worked with summer squash, you know about the irritations it causes. Those same trichomes tend to scratch the fruits during the post-harvest period, which causes a loss of quality. So all of these new summer squash cultivars have this glabrous feature, which makes the leaves and petioles very smooth, with benefits for crop quality and the people working with these plants. So as I mentioned, cucumber has already become a major CEA crop, but summer squash has not. And so why is that? And some of that comes down to breeding, because in cucumber, breeders have released varieties of cucumber that are what we call gynoecious, which means they only produce female flowers, which are the ones that give you fruit, and they are also parthenocarpic, which means they don't need pollination and fertilization. So that's a big topic when we start to try to produce fruit crops in controlled environments, is if they need pollination, then we need to introduce something like bumblebees. And so that's part of what's enabled cucumber to take off, is they have these varieties available. And cucumber, as I'm sure you know, has this vining habit, which enables them to climb and really use the space available in a modern, tall greenhouse structure. And as I mentioned, most of our summer squash tend to be, we've been breeding them for years for field production and breeding them to be very compact, and that does not effectively use the space available in greenhouses. There are a handful of parthenocarpic zucchini on the marketplace currently, and we're actually testing those right now. They seem to be not 100% parthenocarpic, and still some room for improvement there. And we hope to get those parthenocarpy genes from zucchini into the summer squash group, because summer squash actually has a much higher yield potential than zucchini. So for a summer squash, we would like it to have attractive fruit. We'd like it to have this semi-bush habit it be parthenocarpic, have it be resistant to powdery mildew, and be high yielding. And if we can achieve all of those goals, maybe we'll see summer squash start to take off as a CEA crop. Those of you that have grown squash here in Georgia, you know we have lots of insect pest pressure. And most of those insect pests we can exclude, right, because they're fairly large. Squash bugs, vine borers, spotted and striped cucumber beetles, we can physically exclude those, but we still have these other diseases that we need to be looking out for. Powdery mildew, downy, and fruit rots. And one of the other areas of research that we're currently pursuing in my lab is around this, what's called Canophora fruit rot. It's a relatively new fungal pathogen. Takes your nice summer squash and turns them into that black, grayish mush that you see at the bottom of the picture. We've been having some challenges with this in the greenhouse. Not a lot known about it yet. Has a wide range of hosts, and we don't have genetic resistance to this disease available yet. So that's one area that we're pursuing in my lab. We're also working on powdery mildew resistance breeding. As I mentioned, currently varieties that have resistance really only have tolerance, so you still will get some disease on the plants, and we only actually have one gene that's been deployed so far. That's called PM0, and plant pathologists will tell you you're in a dangerous place when you only have one source of resistance, because that can break down over time. So we have some new sources of resistance. I did some of this in my dissertation, and we're still trying to understand more about them. We want to map them to the genome and develop molecular markers for them. And in the future, we hope to be able to stack multiple resistance genes, which we really need those markers to be able to do that, and transfer these genes across the different species of cucurbita, which is manageable. We also want to make sure that our resistance holds up against different races. Powdery mildew has multiple races, and we need to make sure that new resistant types actually hold up against different isolates or races of powdery. So another area, melons, mostly muskmelon, cantaloupe, honeydew, these types of fruits are starting to be produced in CEA. And there's this opportunity, I feel, because we can really make sure we produce a really high quality fruit by being able to tightly control the environment. And so this is a second crop group that I'm starting to work on, are melons. One of the challenges with melon is that melon actually sets the fruits out on lateral branches rather than the main stem. So it makes the management of those plants a little bit trickier, whereas squash plants, we can actually prune off any laterals. If we prune all our laterals off of melons, in most varieties, we won't get any fruit. We're starting to see what's the fastest growing part of the melon industry currently are small, what people are calling personal-sized fruits. And the nice thing about those is if we have them in the greenhouse, they're lightweight enough that they don't have to be suspended in a bag. As soon as the fruits get larger, typically they do require suspension, which is another aspect of labor and cost. We plan to do some work around crop load management, because if you allow the melon plant to set all the fruits naturally or let it fully set, say, up to 4 or 5 fruits, it may not be as good of quality as if you have just 1 or 2. Melons do require pollination, so we don't have anything really out there yet to work with parthenocarpy. again, need that resistance to powdery mildew, and we'd love to see some melons that maybe have a slightly more compact growth, and if we could get them to fruit on the main stem would be really great. So with melon, there are lots of commercially available molecular markers currently, and so that helps us out a lot. And as I started my program, I was able to send seedlings, some leaf tissue off to a commercial lab and have them screen them for important traits such as fusarium resistance, powdery mildew resistance. They can even tell me what flesh color that melon's going to have if I'm generating different colored flesh melons. And I think, we really just see in our markets cantaloupe, and honeydew, and watermelon. But globally, there's a great diversity, including that pretty striped melon. That's an Indian melon called Kajari. The yellow ones are Korean melons. On that bottom left are the Japanese melons that are given as gifts in Japan. It looks like a cantaloupe, but it has green flesh, kind of, if any of you have ever had a Gallia melon. So, I'm running out of time, I know. I just want to mention the third crop that I'm starting to work with are tomatoes, right? A firmly established CEA crop. And we have, this is something where, as Dr. Ferrarizi was talking about, these vertical farms, you really need ultra-compact plants. And so there are some highly dwarfed tomato varieties, but the fruit quality is not quite there. It's not amazing. And so I'm working on making some crosses between these ultra-compact ones with, say, heirloom types that have the improved flavor profile. And again, working with disease resistance genes, we still need to have our disease resistance, and my postdoc currently is working on a project on the tomato yellow leaf curl virus, which is a whitefly vectored virus, quite problematic to the tomato industry. And I'm a big believer in breeding as a team-based approach. I don't really think it's something that one person can handle, and so it requires collaboration with, say, pathologists, with food scientists as far as testing flavor and testing consumer preferences. We have to also not only breed the cultivars, but also generate the cultural practices needed to produce these things profitably. So the end goal then is that we have new cultivars and associated cultural practices. And fortunately, we have, as I mentioned earlier, lots of great modern tools at our disposal. And I am using some digital tools, a platform called BreedBase, where I'm able to store all of my breeding lines, and an app called FieldBook that enables us to take our data directly into a tablet and get away from all the paper data management. So I will wrap it up there. Do we have any time for questions? Or we? One or two questions. Okay. Anybody have questions? Yes.
Are you looking at downy mildew resistance as part of your treatment?
You know, I have not started to work with downy yet. My experience has been with powdery mildew. But it is something that's on my radar, and I think we will, ultimately prior to release of cultivars, want to screen them for downy resistance as well.
Okay, I'm in a cucumber producing area, and we've had scenarios where downy carries over into the greenhouse and then establishes the problem early in the season in the field. It can be devastating because the greenhouse then serves as a carryover reservoir for downy, resulting in early infection production.
Okay, yeah, I mean, the same thing does occur with powdery mildew. In areas where it normally would not overwinter, but it stays alive during the winter months due to greenhouse production. Where is this? Are you located? Eastern North Carolina. Great. Thank you. All right. Thanks. Thank you. Appreciate it.
Up next, the title is Vegetable Disease Management Under Controlled Environment Agriculture, Management of Foliar and Root Disease of Vegetables in Controlled Environments is going to be discussed.
All right. Good afternoon, everyone. My name is Jorge Reyes, and I am a postdoctoral associate in the Department of Plant Pathology. And I work for Dr. Ruchika Kashyap, and she was supposed to be here this afternoon to talk to you about this talk, but unfortunately, she had some other conflicting commitments, so I'm filling up for her, so I'm going to talk on her behalf. And I'm going to be talking about vegetable disease management in CEA, and we'll give you some highlights of the work that we have been doing in the past 6 months. So, yeah, as you know, global population is growing, and there is a huge demand for food production. But beside that, there is, of course, the need for locally grown products, fruits and vegetables. And, and CEA seems to be one of the answers for these type of things because we can produce food year-round and without worrying about the outside weather, right? So we can classify CEA. I know we have been hearing about CEA this afternoon from our colleagues that previously talked, but we can classify CEA about the cultivation method and the cultivation format. If we think about the cultivation method, we look at soil or substrate cultivation, hydroponics, aeroponics, aquaponics, and when we look at the format, we can think of infrastructure, the greenhouse or vertical farming. So this figure was shown previously as well, and this is just to illustrate that we have several big companies that are producing leafy greens and tomatoes and some other specialty crops here in Georgia, and they are very important, right? But besides these big companies, just like Andrew said as well, we have some other upcoming small and medium growers working in the urban and semi-urban areas in the state as well, and they have relatively lower technology probably than these big companies, but they are still betting and trying to invest in crop production as well in these controlled environment facilities. We have the substrate-based systems, and we also some hydroponic systems, just like deep water culture, tower production, NFTs, and even microgreen productions in this type of shells. So there is a lot of ongoing research in CEA. We look at different things as crop production and CEA, light supplementation and light optimization. There is a lot of work being done as well in sensor utilization and automation. But we have to keep in mind as well some of the examples that Andrew showed. The plants get sick as well. Just as in open field productions, our plants in CEA get sick. And of course, with these disease challenges, there are needs for plant disease management. I know that probably you are all familiar with plant diseases, but I just want to briefly mention that a plant disease is the impairment of the plant's normal functioning, and this affects morphology, physiology, and consequently, this is going to affect harvestable parts, especially like if we talk about leafy greens or some other type of fruits. And a plant disease results from the continuous irritation from a pathogen, right? It usually is progressive over time, and it leads to the development of symptoms. When we think about the factors affecting plant disease, us plant pathologists cannot help but think about the disease triangle that we are taught. And we need three things for disease to happen. We need a host, of course, a pathogen that can cause disease, and the environment that conditions that relationship. The host is your crop, so you are going to have the host of the disease triangle, of course. But it is important to mention that even if you don't have a resistant cultivar per se for a specific disease, a healthy plant is more tolerant or resistant to a pathogen than a stressed or injured plant. So just a couple of things to consider. And in the case of the pathogen, Also, we not only have to have the pathogen present in your system, but also it has to have the capability to cause disease. And many of them are host-specific. And in the environment, as I said, this pretty much condition the relationship and the interaction between the host and the pathogen. And in a greenhouse or an indoor production facility, you can get a high humidity, for example, just from irrigation, right? You can get wet foliage or wet substrate. If you have poor air circulation due to high density planting or light intensity factors, that can influence that relationship as well. And of course, if these things are not controlled properly, you are going to have disease happening. So, but if we learn of these type of things in our specific system and in our specific crop, we can use this information to manage disease, right? So, in the case of CEA, some of the important considerations and practices that we can take, that we can look at, for each of the sides of the disease triangles. For example, the most desirable things for for crop production is having resistant or tolerant cultivars, right? So if you have resistant cultivars available, just pick those, right? But if there are not resistant cultivars, try at least to get disease-free planting materials or get your seed from a reputable source. And in that way, you are already attempting to manage disease. From the pathogen side of things, we can look at ways of how to prevent the entry of the pathogen in your system in the first place, right? We can look at sanitation. We can try to disinfect our tools and surfaces. We can try to stop the spread of the pathogen by disposing of diseased plants, right? If we need to identify those pathogens, do a proper diagnosis to do corrective actions. If we have to use fungicides or some biofungicides, let's use them. But of course, you have to know that what is being labeled for the use in CEA as well, because that's another situation. Just because you use a pesticide out in the field, it doesn't mean that you can easily use it in an enclosed facility such as these settings. And in the case of diseases that are transmitted by vectors, such as viruses, for example, we have to control the vector as well, the insect vector. And for the environment side of the disease triangle, we need to find ways to optimize that environment for the benefit of our crop. But how can we modify that environment against our plant pathogens? And that way we can manage disease. So how do we fit in this case? So the recently created Urban and Control Pathology Lab is led by Dr. Ruchika Kashyap, and she is part of the CEA cluster at UGA as well for research and extension. And the idea and mission of the lab is to create an integrated disease management system, that is going to be system, crop, or practice specific, so we can generate solutions to understand the pathogen problems and the needs of our stakeholders. How can we help our stakeholders with disease, pretty much in general? We will be doing research in different systems and in different pathosystems. And of course, with our results, we will look to disseminate these to inform our stakeholders, our growers, and how to combat and manage disease. So in the first 6 months, since the lab was created, we have been conducting some surveys across the state, and we have, number one, identified the type of systems that we have, of course, in CEA in Georgia, but we have also been able to identify some of the major problems and needs that there are for us to work. So these are just some of the examples of the facilities that we have visited. We have some deep water culture production, for example, in White County. We have some tower systems in Forsyth. We have some Dutch buckets, which are substrate-based as well, systems in Gwinnett. We have some hybrid systems, and again, some substrate-based systems. And as I initially mentioned as well, we have some growers producing microgreens as well. We will be interested in looking at and working with growers with high tunnels. We have actually visited a few as well in Athens and Oglethorpe County. And back in August, Dr. Kachap was also surveying and visiting some of the community gardens across the metro area in Atlanta. And the idea of visiting these gardens was as well, look at what type of systems there are, and what are the needs and major problems that our growers are facing as well. So with the pathogen challenges that we have found some root rot causing pathogens. We have oomycetes such as Pythium and Phytophthora. We have Fusarium, and we have some other pathogens such as Botrytis, powdery mildews, and downy mildews, being a big problem in these facilities. In the left, you can see, for example, we have the typical root tip discoloration in lettuce and in basil. This is caused by Pythium. This is just to mention a few examples. And we have as well in the middle image, we have some fungal pathogen called Plectosphaerella, which actually hasn't been officially reported in Georgia. And we found it in one facility. We don't want to, create, we don't want to get people paranoid or anything as well, but this is a pathogen that we have been seeing a lot, actually, in basil and in lettuce. So we are actually doing some work with that, some pathogenicity tests, and to make sure that this pathogen is actually the one causing these problems. We also have in the far right poor seed germination caused by Pythium as well in some spinach seedling production. And of course, we have found some of the old traditional problems, such as anthracnose, bacterial root rot, powdery mildew, and in the far right, southern blight, which is caused by Attilia rolsi. So what do we want to do with this information? And and I just want to mention some of the ideas and the work plan that we have and things that we want to look at. We want to work on hazards evaluation, for example. We want to look at different entry points of these pathogens into these systems. We want to look at how the different factors or elements work as a whole to condition the disease development. And with this, we will hopefully determine critical control points for specific diseases in CEA. Besides entry points, we will also be looking at how these pathogens disperse and spread in the system. What is the role that people are playing, air or water, and some other mechanical actions that happen in the facility as well. How are they influencing the pathogen spread? And of course, the other environmental factors that are very important as well, as I mentioned before, that condition the interaction between your crop and the potential pathogens. So the strategies for a system approach in terms of disease management, we will be looking at from the prevention side of things, what can we do to prevent disease? But if a disease is present as well, how can we do a proper diagnosis and then potentially a corrective management action as well? So some of the ideas, just to be brief, we'll be looking at some cultural management practices. We'll be looking at sanitation, materials, water treatments, irrigation type, and we'll be doing research on these type of things to try to find answers for the main problems that our growers are facing. In terms of diagnostics, we'll be looking at determining critical control points for major plant pathogens. We'll be developing disease diagnostic tools, and of course, using the ones that are already available. And we are also looking at the possibility of working with AI and plant disease imaging, and this, again, with the idea of diagnostics and monitoring of plant disease in CEA settings. And in corrective management, we know that we have several options with this, especially when we look at chemicals, but we also want to look at biological control management. We want to identify chemical alternatives, test different product efficacies, and we want to look at volatile organic compounds, and we'll also be evaluating the effect of other type of stressors as well. And with this, we expect to get some sustainable solutions through collaborations with our partners and different collaborators, such as crop specialists, engineers, and of course, county agents and growers that, of course, have to be part of this. So finally, I just want to introduce what is the lab vision, which is working together. Basically, we want to develop our research hypothesis involving local knowledge and the involvement of our stakeholders so we can foster collaborations. We need and we want to prioritize the needs again of our growers so we can actually execute these projects and do this, of course, in a cyclic way, so with a hopefully evaluation and feedback system so we can have results for you guys and be able to inform and disseminate through a strong extension program. So this semester, we will be launching our website, and this is going to be a scientific hub in which we have different type of extension publications, research publications. We'll even have a podcast. Dr. Kashap is working on a podcast which is going to be called Urban Pods. And this is going to be to have information, of course, in audio, so growers can be even working out in the field and listening to a podcast with interesting information about disease management in CEA. So be on the lookout for these things. And yep, that's it. If you guys have any questions, I'll be happy to answer.
Alrighty, up next, we have Dr. Eric Schueller with the University of Georgia. You can.
All right, good afternoon, everyone. Last but not least, I'll be talking about pest management and controlled environment agriculture. So I'll sort of be splitting my talk into two parts. The first is sort of describing some of the unique pest management challenges that we have in controlled environment agriculture, and then talking specifically about biological control options that we have for some of our major pests in greenhouses and other related structures. So many of you have probably heard the term IPM before, but might not know exactly what that means. Well, simply put, integrated pest management is a science-based decision support system which aims to reduce the use of plant protection products to control pests to ecologically and economically sustainable levels. And IPM is a multifaceted approach. So you can see here, I'm going to take this. Okay, there we go. You can see here with this, this triangle that it's showing sort of the most important components of IPM from most important to least important parts that, we don't want to have to get to the top of the pyramid here. So at the base is prevention. So prevention is the best medicine you often hear. And this can include things like using resistant varieties to pests and doing crop rotation. Also very important is detection. So making sure you're constantly out there doing surveys for pests and catching them at very early stages so you can get them at points where you ultimately don't have to do severe control methods. If you ultimately have to use control of pests, then using mechanical control methods such as removing infected plants, doing pruning, making sure that you're sanitizing the greenhouse is a very important thing. If that's not enough, then moving to biological control, which is the use of natural enemies or predators, which are parasites, and releasing those to manage pests. And ultimately, as a last resort, turning to chemical pesticides, right? And so IPM is a sustainable method in order to manage pests, which is synergistic with the goals of controlled environment agriculture, which also revolves around sustainability. So sustainable local production, the ability to do year-round production, achieving higher crop yields, lowering inputs in water and energy, and implementing tactics which don't leave residues. And so this is where IPM falls into. And then finally, creating predictable quality and yield of the crop. So IPM is a very important component of controlled environment agriculture's goals. Due to the constraints and time I have today, I'm really only going to focus on biological control options. So you already saw this slide in the last presentation. I've modified it a bit here. But it's just to bring home the point that controlled environment agriculture is a really diverse cropping system. You have everything from low-tech, high tunnels and greenhouses all the way to very high-tech vertical farms. And with each of these cultivation formats, you have different cultivation methods, ranging from soil-based techniques, which you typically, you see crops grown directly in the ground in high tunnels. You also see crops grown in soil in pots in low-tech greenhouses. But as then we move up these technology spectrums, now we start introducing hydroponics and hydroponic systems that are not only horizontally produced, but also vertically produced. So all these create very diverse environments to have to control pests, right? So just to kind of bring that home a little more, so here's kind of a cartoon of a vertical farm, right? The crops are grown in vertically distinct layers in the greenhouse, and this can create really interesting effects on the ability of pests and natural enemies to establish in the crop, right? So when you have vertical layers, that can impact the ability of pests and predators to move not only horizontally through the crop, but also vertically through the crop. So in a traditional greenhouse, they might just move across the bench surfaces, right, pretty easily, but in a vertical farm, they might not have that capability. And this vertical nature also introduces other variables such as light gradients, humidity gradients, and temperature gradients, all which impact insect populations. And it doesn't necessarily have to be a vertical farm. So here you can see some indeterminate tomatoes, which are 30 feet tall, right? This picture clearly shows you that, within that crop canopy, there's a big gradient difference in light intensity, and you would expect there to be temperature differences and humidity differences too, right? So the crop is not a uniform environment, which can create really interesting pest management challenges. All right, so a lot of what we as researchers have developed for you over the years for vegetable management in greenhouses has looked at systems like this, or systems on a benchtop, a very uniform, even environment. Well, now we have situations like this, right? So how are the things that we've learned over the last decades for managing pests in the previous situation applied to this sort of situation? And to be honest, it's sort of a new frontier. We have to relearn a lot of the things that we've developed to be able to manage pests in these new emerging high-tech systems. So just for an example, imagine that you have a whitefly outbreak in your hydroponic lettuce, and they are distributed vertically through that system. Well, traditionally, the way that we manage these pests is with biological control, is through the use of a combination of parasitic wasps, which can obviously fly, or predatory mites, which are only able to crawl through the crop, and we'd use those as a pair, right? They're more effective together than alone, and we rely on that combination to be able to eliminate pests. Well, in these vertically constructed systems, you might imagine this crawling biological control agent is no longer effective. It cannot physically get to the upper layer of the crop to manage that. So this completely throws a wrench in a lot of the things that we've developed as biological control systems to manage pests in these systems. All right. So I talked a little bit, sorry. What's going on here? Oh, different button. Okay, back to where I want to be. So I talked a little bit about the structural complexity of controlled environment agriculture facilities being a problem for creating variance in the environment. So another big thing that growers of CEA do is use supplemental lighting. That is very important to achieve the goal of CEA, which is to maximize yields, right? So they rely heavily on supplemental lighting, often in the form of LEDs. This lighting is often very precisely controlled, not only for its intensity, but the wavelengths that we're selecting to maximize crop yields, right? So insects are very sensitive to light. And so the lighting that we're using in these facilities can have a direct impact on their ability to grow their populations and establish on the crop. So just one example here, looking at a predator. So this study looked at the ratio of blue light to red light that was being used to grow lettuce. And what they found was as the ratio of blue light decreases in the system, you get more and more male wasps being produced in the population. Now, why is that a problem? Well, it's the female wasps that are attacking their hosts, laying the eggs in them, so that basically the males have no ability to control those pests. So it's problematic from a pest control standpoint to have a facility that has a bunch of red light, because you're going to get a bunch of males and they're not going to be able to do anything for pest control. So that's something you need to consider as you're trying to grow these crops, is how is the lighting you were using impacting pest populations. So just another example. So that was an example on a predator. So here's an example on a pest. So growers use far red light a lot in lettuce production, because that far red light elicits a shade response in lettuce, and that shade response in lettuce is higher leaf area, right? And that's the thing that we're eating, is the leaves of lettuce. So it can have a really big impact on improving yield. So you can see for this picture here, as the far red percent increases that we're putting on the plants, you get bigger and bigger leaf area. Well, when a plant devotes more resources to vegetative growth, it has to take resources away from something else. And often the case, that is secondary compounds. And a lot of times those compounds are defensive compounds against herbivores. So this study sort of shows you this fact. So this is a study that looked at the effect of far red light on spider mite populations. And they saw as you increase the far red light, those spider mite populations explode. And that's because those plants are less defended against herbivory, right? So those are some, I just wanted to sort of get in your mind really quick some of the unique challenges that we see in controlled environment agriculture due to the new production practices that we're implementing, and how those are potentially impacting the ability to control pests in that area. There's a lot more, but I think those are some of the more interesting ones. So we could talk about other things later if you're interested. But for the sake of time, I want to move on and talk about biological control in CEA. So the first group I want to bring forward, hopefully most of you are aware of these, is whiteflies, which, as the name suggests, is an insect that kind of looks like a white fly. And these can reach really, really high population levels. They are feeding on the phloem tissue of the plants, and as they're feeding, absorbing the sugars, and then they excrete those sugars as they're feeding and create a big sticky mess on your crop. And that sticky substance called honeydew can promote the growth of molds, which reduces photosynthesis and decreases plant vigor, and it causes a whole bunch of other problems. So how do we manage those with biological control? Well, we have a lot of options. So a lot of the things I'll talk about have sort of cross-use through other pest groups, but I'll introduce them as I go along. So there's other things besides this that work for whiteflies, is what I'm trying to say. You'll see those a bit later. One of the biggest tools we have is parasitic wasps. So the two that are commercially available that we use a lot are Eretmosferus americus and Encarsia formosa. Here, that's Eretmosferus, this is Encarsia. Again, both commercially available. You typically have to purchase both of them to control whiteflies, because they do well on a particular whitefly species. So both sweet potato whitefly and greenhouse whitefly, if you have whiteflies, you probably have both of those. So you're going to need to release both parasitoids. We also have predatory beetles available. These are from the genus Delphastus. These are all native here in the southeast. I put an asterisk next to Catalinae because this is the only one you can buy commercially. But if you live in an environment with vegetation around you, you often will see these other two get into your greenhouse and sort of naturally control your whitefly, if you're lucky, that is. So how do we use these parasitoids? We typically buy them using these cards, which have all these infested, parasitized whitefly nymphs, and you hang those in your crop, and then the adults emerge out of those cards. It's very important whenever you buy biologicals from a vendor that you test the quality of the product you're buying so that, say, if you're not working, you can blame it directly on the product efficacy. In the case of whiteflies, it's just a matter of putting those in a jar with some tape, and making sure you're seeing those adults emerge as they're coming out. If they're not emerging, you've got a bunk product, and you need to contact your vendor. There, right? And you typically are releasing a 1-to-1 ratio of Encarsia to Eretmoceras there. You don't have to release them that way. You can use a banker plant system. And a banker plant is simply a non-crop plant that you're putting adjacent to your crop. And it's providing a resource that your crop is not for those natural enemies. So this is a papaya banker plant system. So here you've got your papaya. And what the papaya is doing is it's housing populations of papaya whitefly. And that is a species-specific whitefly. It will not attack anything but papaya. So you get super, super high levels of papaya whitefly, and that's building up massive populations of your parasitoids and beetles. Those are moving into your vegetables and controlling your pest whiteflies. So the next group is aphids. Aphids are a really nasty group because they reproduce very, very quickly. And the reason for that is they have what's called telescoping generations. So essentially, the grandmother is pregnant with the mother, which is pregnant with the daughter simultaneously. So when the grandmother gives birth, the mother is already pregnant. So they're extremely difficult to control. And much like the whiteflies, they're feeding on sap and causing a mess. So how do we control aphids? Much like we control whiteflies with parasitic wasps, we use a different genus. In this case, we really like to use the genus Aphidius. Aphidius urvi, which attacks big aphid species, and Aphidius comini, which attacks small aphid species. So you need to know what aphids you have to be able to select the right Aphidius control agent. Again, these are commercially available, and if they're successful, you'll start seeing these aphid mummies being produced. You can see the holes where the adult parasitoids are coming out, and then they come and lay eggs in other aphids. When you buy them like that, they typically come in a shaker bottle with a bunch of mummies, and you just spread the mummies out on your crop. But you can also use banker plant systems for these guys. In this case, you're using another specific, so this is an aphid that's specific to grasses called bird cherry oat aphid, and you want to infest them with something like barley. Put that in your greenhouse. You can see them hanging out on the post here, and that's going to produce large numbers of Aphidius, which are moving into your crop and controlling the pest. Thrips are really nasty. Unlike the aphids and whiteflies, they're chewing damage. You can see the very characteristic sort of scarring on leaves. They really like to live in flowers, so if you're scouting for thrips, make sure you're checking the flowers carefully, because most of them are often in the flowers. We control these typically with what are called predatory bugs. And these are really great, but they're challenging because they require lots of resources to sustain themselves. So Aureus insidiosus here and Disophis hesperus are commercially available. You can release them in jars, but you can also get them naturally from the environment. They're pretty widespread in the United States. They also have, so if you're releasing Aureus, it's really important to have flowering plants in your greenhouse. If you're growing a crop which does not have flowers, you need to provide them, such as alyssum. And for the Disophis, they love mullein. So you can see a tomato grower that's put mullein plants all along their greenhouse, and they feed on the mullein sap and then move into the crop to control your pests. And these are often work best when they're used with other things like parasitoids and predatory mites. Just to wrap up here, spider mites, everyone hates spider mites. They're very difficult to control with pesticides because they produce webbing which protects them from pesticide residue. And they produce this very characteristic mottling on leaves, which basically turns the leaves to paper after they're done feeding on it. We like to use predatory mites for these. The king of predatory mites for spider mites is Phytocoelus persimilis. It can handle the webbing that spider mites produce. Other mite species struggle with that, but I wanted to include them here because, as I mentioned, there's a bunch of things that work for a lot of different pests. And so for whiteflies, you have Amblysteus swirsky, and for thrips, you have Neosteolus cucumeris. They're mainly being able to deal with the immature stages. Once they reach adulthood, you have to use something else, like the predatory bugs or beetles. And predatory mites, typically sold in these sachets and bran. You can either hang these from the plant or spread that bran across the crop. And then finally, the last group I'll touch on here briefly is fungus gnats and shore flies. I'm only including shore flies here because they look a lot like fungus gnats and they're in the same sort of conditions as fungus gnats. But shore flies do not cause plant damage. They are strictly an aesthetic pest. Fungus gnats, on the other hand, will eat the roots of your plant. They thrive on very high humidity environments. They're basically semi-aquatic, right? So the best way really to manage them is through water management, not letting your plants get too wet for too long. But if you have to manage them, you can do that with biocontrol. So just to talk about these shore flies quickly, the main thing you have to manage them is right before shipment of your product, because if your customer is getting a plant with a bunch of flies on it, they're probably not going to appreciate that. But otherwise, they can just be in your greenhouse and they're not doing anything. So how do we manage these flies? Well, we have entomopathogenic fungi, which is basically a fungi that inserts itself into the white fly, or to the insect, develops in there, and it kills it from the inside. A bunch of different products available for that, and we usually use entomopathogenic fungi as a foliar spray, but I really think it works best as a dip. So if you have chronic fungus gnat problems, dipping your seedlings is a really good way to keep that from being a problem for most of the production cycle. And I think I'm right on time to finish here. So I'll be happy to take any questions. I'll leave this slide up since I didn't have time to get to it, but nematodes also work very well for those shore flies and fungus gnats.
All right, thank you. Here, this is for you. And your phone, is this your phone? All right, that is the end of the controlled environment session. Thank y'all for coming.